देखो फिर मेरी इच्छा कैसे रहेंगी, फिर ये है मेरी इच्छा। हाँ, आपने ही बोला ना, मैंने तो बोला ही नहीं। ओके चलो, so let's start the lecture. So already we are started the lecture on unit number 3, a scale up process. Last time we are discussing the certain important issues regarding the isolation. After the isolation, we will switch over for the screening of the organism. It is the fundamental and very essential task one if you are an industrial microbiologist, because day by day the human consumption, it can get increases one, and because of the what you call them as in the cement trees are there in the form of the big towers, big flats systems are there. The cultivated land day by day it can get a decreases one. Now this is an alternative mechanism how to increase the product. Okay, so either for the primary metabolites or either for the secondary metabolites. So screening is always followed by the two important process, primary as well as the secondary. And after the secondary screening, it is your moral duty to preserve this culture for a longer period of time, depending upon which type of these industries are there. Okay, if my industry, it is manufacturing the any kinds of the food, so according to that, I will maintain my isolates one. If it is an pharmaceutical industry, so according to that, it can get a preserved one. But the preservation mainly of these 6 important way. Okay, now what exactly that maintenance of the isolates? We will go with the first in a brief manner introduction. The maintenance of any type of the pure culture for the extended period only in the viable condition. Your microorganism should be a viable when it is get a preserved one, but at the same time it without any genetic change, which is simply referred as in the preservation, or we call them as in the maintenance of the isolates. During my preservation, most valuable a microorganism, you have to firstly stop the growth of a microorganism, or you have to slow down the growth rate. So whenever we are preserving the culture, definitely there will be the growth, but the rate of the growth, it is very less one as compared to the normal, which is get incubated in the incubator at 37 degrees Celsius. Due to this toxic chemicals are not get accumulated, and hence the viability of the organism is not get affected. So these are the certain objectives in my way how to preserve my culture for a longer period of time. If you cultivate the microorganism and preserve in the form of the master plate, now that master plate, it can get preserved for the 1 month, it can get preserved for the 3 months, it can get preserved for the 6 months, and the same culture, it can be preserved for the years also. But during the growth, most of the times what's happened, the organism liberates the toxic chemical, and that toxic chemicals, they are with a responsible one. Okay, and in this preservation technique, there should not be any accumulation of such type of the toxic chemical. Otherwise your microorganism may a chance there should be either killing or either they undergo for the inhibition process, and we do not want during the maintenance of the isolates one. Okay, now what are the application of these preservations? When I am a teacher, I will use as in the academic use for day-to-day practical work, either for the semester 3rd, either for the semester 5th, or either for the semester 1, or for the PG, that will be the 1st, 3rd, and their research activity also. But if any one of the student who are involving in the research activity, so the preservation is also being done for the research purposes one. What exactly happening? Whenever XYZ type of the chemicals we are get utilized one, what will be the effects on the growth of such type of the organism? So at that time, so you have to use for the research purposes one. Just now we are doing this. Most of the time the pharmaceutical companies, really their antibiotics, they kills the microorganisms or not. So it is one part of the research activity. So we call them as the R&D, research and development, where exactly your product, it can get a tested one through the toxicological way, and through another by the biological, through by the microbiological testing one, either in the form of the assay test are there. So either that will be the qualitative as well as the quantitatives are there. And the preservation is also being used in the fermentation industries. It is very important because day to day, entire world scenario, it can get a changes. If you think near about 3 decades before, nobody looking for the fermentation techniques. Why? Because at that time, neither any of the sophisticated instrument informations are there, and we are doing all this activity with the help of your conventional techniques as well as the traditional way. Whatever maybe the synthesis are there, so according to that we have to add the A as well as the B, it can produce in the D1. But the today's scenario in the industry, it is totally get a changes one. Now everything we are looking for, every product for utility of the what you call them as in the mankind, it is totally depending upon the microorganism right now. Each and every the product right now which is get launching into the market, it always comes from the microbial biomass. Either it may be the single cell protein, it may be the antibiotics are there, it may be the steroid, it may be the hormones, it may be the vitamins, it may be the acid, it may be the biofuels are there, it may be the mining industries are there, etc. and etc. So everywhere we require the microorganism, and for that the preservation is the good art to maintain this what you call them as the economic, valuable, incredible microorganism in a different way. And now the today's era, we are facing the large challenges against the COVID-19 coronavirus. Really, whether there will be the medicine or not. So where this particularly the testing, and this is testings on the microorganism, testings on the animals, testings on the human beings later on. So this is, we call them as the biotechnological field, where the large branches it can get explore one. Because of your microorganism, we are maintains one for a longer period of time. Preservation of microorganism, what exactly that? It is one of the vital, important step, phase in every fermentation process. To preserve the isolated in the original forms only. And the preservation, we are done in two methods. One we call them as a continuous metabolism, and the second one we call them as the suspended metabolism. All of you know what's happened when the milk, if it is not get heated and put at a room temperature, what will be the shelf life of that milk? It can withstand with the either the 6 hour, or at the max that will be the 12 hour. And after 12 hour, your entire milk, it can get a spoil. Okay. Now, and take the same milk sample with a pasteurization temperature, What will be the shelf life of that milk? ये हमने देखा है बहुत बार, हमारे घर का जो दूध रहता है वो कभी कभी फट जाता है। फट जाता है यानी क्या होता है मुझे भी नहीं समझ में आया। और जब मम्मी उसको बॉईल करती है और रख देती है तो एक दिन भी चलता है, 2 दिन भी चलता है। I do not know what exactly the happening, but it is a part and parcels of the preservation technique. Here we are interested in the preservation of the microorganism, and your mother is interested in the preservation of the milk sample. They destroy the microorganism. Here we are maintaining the microorganism, either by the continuous metabolism or either by the suspended metabolism. Okay. Now, maintenance of your isolates through the continuous use of the microorganisms, especially that will be the specific type of the isolates, or you call them as in the strains one, which is generally used for the research purposes, assay development is there, or basically for the production of the XYZ product, we call them as in the production culture. Now, all these strains, they are highly valuable. They are very important and must be maintained or preserved over a long period of time without any genetic as well as the phenotypic changes. Phenotypic changes are those one which can very easily seen by your naked eye. What will be the exactly the shape of the colony? If it is get a disturbed one, we indicate that there may be the something goes wrong during the growth one. Either there will be the contamination by the another microorganism, or the microorganism itself undergo for the mutation mechanisms are there, etc. and etc. Okay, so that is known as the phenotypic. Pheno means which can seen by your own naked eye, and the genotypic characteristics are those one which is 100% related with the genes one. Okay, now the very first one, your microbial strain discovered to produce a useful product, we call them as in the research culture. Okay, what is that research culture? So any particularly the strain who discovered to produce the any kind of the useful product, it may be anything, that is known as the research culture. And how a research culture is differs from the assay culture. The organism used for the assay of a biological activity of the microbial product, as well as for the various kinds of the screening techniques, we call them as in the assay culture. Assay means you have to check the quality as well as the quantity. I am using the XYZ culture for my XYZ practical work, but it having the certain biological activity, I have to test it. The any particularly the antibiotic which is available in the market. For example, either it may be the penicillin, it may be the tetracycline, it may be the streptomycin, it may be the erythromycin, it may be the gentamicin, kanamycin, whatever may be the mycins are there. Really that antibiotics, it can inhibits the growth of microorganism. Either it may be the gram positive or either it may be the gram negative. Are you testing? We never testing. Whatever may be the physician prescribe the drugs, we are go toward the chemist, bring it and takes immediately. We are not taking all these efforts because it is not our job. It is a job of the industrial microbiologist, where they are particularly each and every the batch, it can get tested out. Either by the any way, either by the assay way or either by the sterility testing and whatever may be the, which are the standard SOPs are there. The research culture, which can produces the metabolites of the sufficient commercial interest, is used for developing the large scale of production. Okay, I'm interested. For example, I'm interested in the certain types of the plant growth hormones. Okay, it is not easily manufacturing it by the traditional as well as the conventional way. You have to very firstly, water maybe the microorganisms are there. So that microorganism you have to put into the biotechnological sense, mainly for the recombinant DNA technology. Certain genes you have to replace one or addition one. So that particularly the culture, it can get what you call them as in the upgrading one. Now that culture basically do not use the any type of the plant growth hormone, either it may be the 2D or the 2,4-D is there, or either it may be the indole acetic acid is there. So these are known as the certain plant growth hormones one. So when you are added to the plants, so there is the abundant growth of the plant get taking place one, because we are putting the artificial hormones one. Okay, so that culture we call them as in the research culture. Okay. Now the various culture can get maintained for a prolonged period of a time by using one of the best techniques. Already we are discussing about the low temperature, so it is always get preserved. So this is known as the subculture. So the another name for the low temperature, we call them as in the subculturing. And that subculturing, you have to preserve this culture at a temperature of 2 to 8 degrees Celsius. It is 100% a refrigerator. Now the refrigerator having the two temperature, one we call them as in the freezer box, and another rest of the moieties, or we call them as in the empty space, that is known as the refrigerator, whose temperature in the range of 2 degree to the 8 degrees Celsius is there. And the freezer box whose temperature will be the 0 degree, and below the 0 degree that will be the minus 22. Depend upon the what you call them as in the makers. Either it may be the Godrej, it may be the Samsung, etc. and etc. Storage as a lyophilized culture, mainly under the vacuum at low temperature. What exactly we are doing with the lyophilize? During the lyophilization, we can remove the free water which is present inside the cell, which is get present in the spores. So from the spores and from the cells, we are removing the free water molecules, and entire particularly the cell which is get present in the form of the powdery deposit. And that powdery deposit, it is nothing but these are known as the lyophilized culture. Another, the storage of the vegetative cells as well as the spores. Last time we are discussing that will be the liquid nitrogen, whose temperature will be the minus 196, as well as the vapors of the liquid nitrogen. The temperature will be the 163 degree Celsius is there. And the preservation of the lyophilized culture is most widely used technique, method for all the industries, for all the different type of the activities. The air dried, this is very simple one at room temperature on a sterile room. You are doing this activity, and you are taking the culture, inoculated on the surface, put into the incubator, and 1 month you are not seeing the incubator also. So what's happened at that time? So the entire media, it can get a dried one. So this is one of the best technique. Either for the, just last year, we are doing this activity regarding with the fungi cultivation by the slide technique. You are not seeing this slide till to the 3 months. So this is one of the way for preserving the cultures. Either it, sometimes you have to use the maize, sometimes you have to use the rice bran, sometimes you have to use the what you call them as in the wheat bran is there. Now whenever there will be the bacterial culture, this bacterial culture may be remain viable up till to the 70 years. This is the natural phenomena, and most of the time the recombinant bacteria we can get preserved with the help of the glycerin stab. So last time we are discussing, and the temperature for the preservation of such type of the important, we call them as in the temperature minus 70 degree, that will be the freezer temperature. Preservation are mainly by the 4 important techniques. One, the periodic transfer to the fresh media, we call them as in the subculturing. Second one, storage in the sterile soil. Third one, the saline suspension. But this saline suspension is not been a preservation technique. It is for the time being activity. And the fourth one, we call them as in the oil overlays method. You have to add the mineral oil, that is 100% sterile. You have to sterilize in the oven. A temperature will be the 180 degrees Celsius for near about 3 to 4 hours. So that mineral oil you have to first sterilize, and that sterilized oil you have to put over the surface of the growth one in the test tube. So that is known as the oil overlays method. Apart from that, the suspended metabolism. You have to suspend the metabolism, stop the metabolism, storage at the low temperature, freeze drying, and the storage in the silica gel. So these are the 8 different methods which is get available for the preservation. Periodic transfer to the fresh media. As a microbiologist, all cultures are the valuable cultures. For any teachers or any student, if they know the value of their culture, the student do not know the value of their culture, they never see the any type of the petri plates one, those they are get inoculated. This is from the by heart a statement. I seen the student last from their 22 years, how they behave in their laboratory whenever there is any type of their cultures we are get provided from the master cultures. Now the fresh culture from the previous stock cultures, the culture medium, the storage temperature, as well as the time interval at which the transfers are made, which can get varies with the species as well. Some bacterial species required the 8 days time is there. Some microorganism it required the 15 days. Some microorganism it required the 20 days. So that period it can get a varies one. If we today inoculate the culture, incubated into the incubator, if the proper growth is get observed one, the same tube you have to preserve in the refrigerator for 15 days. But after 15 days, you have to take the culture and again transfer onto the fresh medium. So this technique is known as a periodic transfer to the fresh medium. Why we required after 15 days? Because within the 15 days, whatever may be the media which containing the nutrient, so that nutrient it can get exhausted in the form of the carbon, in the form of the nitrogen, in the form of the minerals, or in the form of the vitamins. And must be ascertained before hands. The temperature as well as the type of the media chosen should support at a slower rather than the rapid rate of the growth, so that the time interval between the transfer it can get take much longer as possible as. So that is very important. Many of the common, the heterotrophs, they are remain viable for a several week or the month on the media like the nutrient agar, which containing the peptone, beef extract, yeast extract, NaCl. Now this transfer method have the advantage as well as they have the disadvantage of failing to prevent the certain changes in the characteristic of any kind of the strain, because the species is firstly the genus followed by the species, and the species followed by the strain. Due to the development of the variants as well as the mutants, which is very essential. You have to know each and every a microorganism, their knowledge of the cultivation, either their species or either there will be the strains. Then what are the advantages of the periodic transfer? The very first one, it is one of the very simplest method, and any special apparatus does not require for the preservation. easy to recover the culture at any moment. A disadvantage, a high risk of the contamination in this method, because after a certain time, after the 1 week or after the 2 week, you have to always expose toward the environment during the fresh transfer, and some of the time if there is the entry of the contaminant, so naturally entirely the tube it is get spoiled. It may be a possible to change in the genetic as well as the biochemical characteristics. There may be the possibility, not 100%. So it again varies from the species to the species, which type of the organisms are there. The lyophilization, we call them as in the freeze drying technique. The freeze drying is one of the process or the technique where the water as well as the other solvents are removed from the frozen product via sublimation. You know very well, if the water it can get a particularly heated one, so the liquid it can get converted into the gaseous phase. Okay, so that process is known as the sublimation. Now the sublimation occur whenever there will be the any frozen liquids go directly toward the gaseous phase. For example, the solid is there. If we hit the solid, and that solid instead of firstly get converted into the liquid and directly get converted into the gaseous phase, we call them as the sublimation. And this technique, it is recommended generally for those using the slow rate for the cooling, as this will result in the formation of the vertical ice structure. For example, see if this culture, if we are preserved at a temperature of 0 degrees Celsius, what's happened? At a freezer whose temperature will be the 0 degree, 100% there is a formation of the ice crystals are there, and these allowing more efficient water sublimation from the frozen product. ये हम देख सकते हैं, हमारे घर पे भी ऐसी एक्टिविटी हो सकती है. Okay, the freeze dried product, they are highly hygroscopic in nature. Whenever you are exposed to this culture toward the what you call them as in the Anurag. Okay, so all these are the hygroscopic in nature. When you expose us toward the environment for a long period of time, water may be the environment containing the moisture, it can very easily trap one and must be protected from the moisture during the storage. Under this condition, the microbial cells are dehydrated and their metabolic activity is totally stopped. With this technique is known as suspended metabolism. जब हम घर पे कोई चीज प्रिजर्व करते हैं 1 दिन 2 दिन या 3 दिन के लिए, बेस्ट एग्जांपल दैट विल बी द मिल्क. So every day the milk is can get a heated one. The heating is not that heating as per your mother do. Heating is as per the Louis Pasteur, that is an pasteurization technique. Okay, if you are doing this one, so your milk sample can be preserved up till to the 8 days without any damages one, or the spoiling get taking place. This is one of the way, but there is the continuous metabolism get goes on, but in a slower rate. Here what's happened, when you remove the water molecule, entire metabolic process it can get a held up one, or you call them as a stop one. As a result, microorganisms they go to the dormant state and retain their viability for the years and years. Lyophilize or the freeze dries of the pure culture are then sealed and stored in a dark at a 4 degrees Celsius in a refrigerator for a longer period. A freeze drying method is one of the most frequently used technique by the culture collection center. So last time we have studied the certain culture collection center, either it may be the ATCC, American type culture cultivation, or either it may be the NCL Pune, National Chemical Laboratory is there, or either it may be the USSR, the Institute of Antibiotic Research one. So there are the N number of the well organized the institutes are there. Okay, so advantage of the lyophilization. Only minimum storage. So small ampoule is there. So small storage space is required. Hundreds of the lyophilized culture can be stored in a small area. The small vial can send conveniently through the mail by the postage. So mail is not that electronic mail. This is by the courier. That will be the postage to the other microbiology laboratory, when packed into the special seal made, what you call them as in the containers one. Lyophilized culture can be revived by opening the vials and adding firstly the liquid media. Why we are adding the liquid media? Because we are removing the water molecules. Now you have to add the liquid into the site the cell, but at the same time the cell required the certain nutrients in the form of the media, and transferring the rehydrated culture to the suitable growth medium. So these are the certain advantages. Another that is known as the cryopreservation. So this is an alternate name, the preservation of the culture in a liquid nitrogen as well as the vapors of the liquid nitrogen. Temperature that will be the minus 196 as well as minus 163 degrees Celsius is there. In this method, your interested microbial culture are rapidly frozen in a liquid nitrogen. When you dip the culture, within a few seconds, the entire culture, it can get frozen away. But you have to add the certain stabilizers. We call them as the stabilizing agent. And the name of the agent, it is most popular, a glycerin, or you call them as the glycerol. Nowadays, we are using the, rather than the glycerol, we are using the inert material DMSO, dimethyl sulfoxide, that prevent the cell damage due to the formation of the ice crystal and promotes the cell survival for a longer period of time. Okay, now this liquid nitrogen technique have been successfully with many species that cannot be preserved by the lyophilization, and most of these species remain viable under this condition for 10 to 30 years without undergoing a change in their characteristic. But however, this is one of the expensive method. Last time we are discussing this, particularly the preservation of the umbilical cord. There are most of the time, there are the certain, nowadays we call them as the banks. Okay, so where particularly the blood, the umbilical cord blood, we can preserve up till to the 25 years. And every year they costing near about 25,000 rupees. Because in future, whenever any organs, if it is get a damaged one, from the umbilical cord, we can utilizes that samples and get converted into the another organs one. Nowadays we call them as the artificial. Not necessary every time. We are proving one. So we are making the lungs artificially in vitro condition by using the stem cell technology. So your stem cells, we are get preserved, protected one for 10 years to the 30 years. But the method is highly expensive one. It is not been tolerated by every Indians. The agar plant culture. Okay, so you know very well. all microbiology laboratory preserve this microorganism in a small test tube. And now that slant, it can get incubated at 24 hours or more than that. And later on, it can get preserved in the refrigerator. This culture, we call them as the periodically transferred to the fresh medium. The time interval at which the transfers are made, which varies with the origin as well as the condition of the growth. So which type of the growth it can get a suitable soil. Okay, so in a coming lecture, we will talk about the agar slant culture covered with the oil, or we call them as the oil overlays method, or another that will be the paraffin method. Okay, so I have only the less than 1 minute. It is not possible to discuss all issues regarding with this agar slant culture covered with the oil. So any difficulties for this lecture, you can write in the WhatsApp group, or if you have any type of the problem, please go to the YouTube link and again listen the lecture carefully. So